Good morning and thank you for joining me today on Science Network. I am Annie Davidson and today we are honoring one of the top DNA scientists, Matthew Meselson. Matthew Stanley Meselson was born on May 24, 1930 in Denver, Colorado. He grew up in LA, California and was always interested in science. He had a huge lab and workshop in his family's basement and garage. Matthew studied liberal arts and chemistry at the University of Chicago before pursuing a graduate degree at the California Institute of Technology under Linus Pauling. He became an associate professor of physical chemistry and senior research fellow at California Tech until becoming a Harvard professor in 1960, where he conducts research and teaches genetics. Meselson has received many awards and honorary doctoral degrees, including the Thomas Hunt Morgan Medal for Lifetime Contributions from the Genetic Society of America. Meselson is an authority on chemical weapons and is still very involved with international policy and politics today. Matthew Meselson has been a major contributor to genetics and science in general for over 60 years. While still at California Tech, his thesis project was using X-ray crystallography to figure out the structure of a specific protein. In 1954, he met Frank Stahl, and together they set out to devise a way to prove or disprove Watson and Crick's model of semi-conservative replication. They were a great team, and in 1957, they succeeded in producing experimental proof of semi-conservative replication of DNA. Also working together as part of the phage group, they invented a new technique, gradient centrifugation. This uses centrifugal force to separate molecules based on their densities. Their classic paper was published in 1958, and his experiment has been called one of the most beautiful experiments in biology. Here is the science behind the experiment. They started with the isotope nitrogen-15, which was then switched to be grown with normal nitrogen-14. When they extracted the DNA using density centrifugation, they found three types of DNA, one containing nitrogen-15 and one containing nitrogen-14, and a hybrid containing both isotopes. When the hybrid DNA was made single-stranded by heating, they could show one parental strand and one that hadn't been newly synthesized. So when DNA is synthesized, the DNA double helix splits into two, and each single strand acts as a template for the synthesis of a complementary strand. This is called semi-conservative DNA replication. Meselson showed in the years that followed many more theories in relation to this one. He co-developed a model for recombination between DNA duplexes that guided field research in this area from 1973 to 1983. Meselson also worked on phage recombination, showing recombination results from splicing DNA molecules. In 1960, Francis Jacob and Sidney Brenner came to California Tech, where they obtained from Meselson the data necessary to prove the existence of mRNA. After accepting his position at Harvard in the fall of 1960, he discovered the enzymatic basics of host protection, where the cell recognizes its own DNA by adding methyl groups to it. Foreign DNA will be attacked and destroyed by restriction enzymes, but host or methylated DNA remains intact. Meselson also discovered the process of DNA mismatch repair, which allows cells to fix mistakes in DNA. His current research interest in the area is targeted toward understanding the advantage of sexual reproduction in evolution. There is even a term called the Meselson effect that refers to the process where copies of a gene evolve independently of each other, becoming increasingly different over time. Since 1963, Meselson has been concerned and active with the use of chemical and biological weapons. He has been an expert consultant for a number of government agencies and studied these weapons in warfare. Today we honor Matthew Meselson for pioneering a new way of doing science with Stahl and many others. He came up with his ways to make predictions based on different models and then determine which predictions is closest to the experimental data. The material he used are still used today. His experiment, the Meselson effect, was simple and elegant. A classic much like the man himself, It is an amazing experiment, and I was excited to tell you about it today.